I'm Dr. Michaelides. I'm an ophthalmologist at Moorfields Eye Hospital in London and a scientist at the UCL Institute of Ophthalmology, and I'm a very grateful recipient of an FFB Career Development Award. So welcome to the science session on participating in a clinical trial. We're going to start with some very brief presentations and then turn things over to yourselves to have a question and answer session. Please note that this session is being audio and video recorded. If you're using an assisted listening device, please turn to channel 1. And please don't forget to turn off your cell phones. The speakers for this session are Dr. Jackie Duncan and Dr. Mark Panesi. Dr. Duncan is a professor of clinical ophthalmology, is the director of the Retinal Degenerations Clinic at the University of California, San Francisco. She's expertise in the diagnosis and management of patients with various retinal diseases and has a strong interest in developing imaging and monitoring technologies to better evaluate both the progress of disease and the efficacy of emerging therapies. Dr. Panesi is an assistant professor in ophthalmic genetics at Oregon Health and Science University. He's part of the RPE65 gene therapy trial for LCA, the STARGEN trial for Stargardt disease, and the USHDAT trial for Usher syndrome. He also serves as co-director of the Visual Function Services and director of the Ophthalmic Imaging Services. Before we begin, I'd like to remind you to take the Power of 3 pledge at the booth located across from the registration desk. It's a summer challenge to take 3 easy actions to tell the world about the great progress we're making in vision research. You can start your actions to raise awareness right here at Visions, like create a message in the video booth or share with friends on social media. The goal is to get 500,000 actions this summer, so join us by stopping by the booth, take the pledge, and walk away with a special power pack. So I'm going to turn the floor over to my 2 colleagues.
Great, thank you very much. So I think we're going to talk a little bit today about what it means to participate in a clinical trial and the different kinds of clinical trials and some of the terminology of clinical trials. But I think we'd like to keep this somewhat informal in that if people have questions, we'll have plenty of time for that. And just to get started, we wanted to maybe get a sense of what types of diseases are represented here. And so maybe just a raise of hands how many people here are interested in hearing about retinitis pigmentosa? Okay, big amount. How about Usher's syndrome? Okay. Stargardt's? Choroideremia? Any others?
Conrad dystrophy.
Conrad dystrophy. Any other ones we haven't mentioned?
Bardet beetle.
Bardet beetle. Okay.
These are some we're happy to talk about today.
Yeah, absolutely. So I think the first thing I was going to talk about, and Jackie can also chime in, was just the basic design of a clinical trial and some of the terminology behind clinical trials. And when we think about research, it often starts in the laboratory, and this may be working with cell cultures, but eventually then we'll find some type of therapy that we think may work, and we'll take it to an animal model. And so prior to anything being tested in humans, it needs to be first tested in animals. And this is both to look at safety of a therapy, but also efficacy. And it often helps if there is an animal model of that disease. And for many diseases, such as RP or Usher syndrome, Bardet beetle, we have mouse models of these diseases. And so in the case of gene therapy, we can develop gene therapy vectors and try to treat these animals and see if they have a response. We can then also look at the safety by actually looking at the eyes afterwards to see if there's any inflammation or any other harm caused by the treatment. And this goes for any kind of therapy, be it a small molecule or gene therapy or stem cells. Now, prior to starting a trial, then you have to accumulate enough data, and usually the FDA wants to see it in at least 2 different animal models. And so you oftentimes will look at a mouse model and then usually a larger model, such as a dog or primates. And primates in particular are important because they're the closest to humans, and so in terms of toxicology, the FDA is always very interested in that. Once you have done that, then you have to look at the pharmacokinetics of the drug to see how it distributes throughout the body, and then eventually you get to the point of filing for what's called an IND, or investigational new drug. And only after all of that has gone on, then you get to what is called a phase 1 trial. And the FDA breaks clinical trials up into 4 phases, 1, 2, 3, and 4. So a phase 1 trial is generally considered to be the very first thing, and it's done in a small number of patients, and usually it's looking primarily at safety of the drug. And oftentimes the patients who are in phase 1 trials are very severely affected, the idea being that if something were to go wrong or if the therapy were to cause some damage, that they would not have much to lose. Now, usually, if the success of a phase 1 trial is determined by if it is safe, and sometimes you get some information on the dosage of the drug that you might want to use. But usually we don't see efficacy in a phase 1 trial. Sometimes we do, and of course that is wonderful if we do. As you move on, then you can go to phase 2, which is looking now at a larger number of patients and looking at optimizing the dose that would be used in a larger trial, also looking for signs of efficacy. But it's really the phase 3 trial that is sort of the workhorse of clinical trials. And this is often where you have a large number of patients, but also you incorporate a component of a placebo, where you treat some patients with an active drug and you treat other patients with something that looks just like that drug, but is essentially a sugar pill. And the reason we do that is to get rid of bias, because we, as scientists, can be biased when we look at patients. We may want this drug to work, and that can bias the results. The patient can be biased because they want the drug to work, and they may over-report things to the physician. And so what we do is we mask both the patient and the doctor to what treatment is being done. And that helps eliminate that bias, and that allows us to get more objective measurements. And so the other aspect of a phase 3 trial is that it's often a multi-center trial, because if we can reproduce the effect over multiple centers, we all do things a little different, that gives us much stronger information than if it's just done at one center, where there still may be sources of bias. A phase 4 trial is not something that we deal with as much. That's usually an aftermarket trial once something has been approved, looking at long-term outcomes. Do you have anything you want to add?
That was really good. I just wonder, is there anyone in this room that doesn't know what we mean by efficacy? So with efficacy, Dr. Panisi is talking about, initially we try and see safety, whether the treatment's going to harm the eye. And then later, as we're pretty sure it's not going to harm the eye, we start to look to determine whether the drug does what we hope it will do, whether it's going to have the effect that we want it to, or have efficacy. And oftentimes that is measured a number of different ways. It could be measured by visual acuity, which is sometimes very hard to measure in people with retinal degenerations. People with retinitis pigmentosa have really good visual acuity until many, many years after they've been diagnosed with the disease. So that's not something that's going to be easy to measure as an indication that the treatment has a strong positive effect. Other things we measure sometimes are visual field sensitivity, or side vision, ability to see lights off to the side. And that's another thing that we can measure maybe a little more readily than visual acuity in terms of change over time. But that also changes pretty slowly, making it difficult to identify significant changes. So people are looking at new ways to measure whether treatments are effective or have efficacy, in addition to looking for side effects or evidence of any safety concerns. So I think all the things that were just mentioned are all really important parts of the different phases of the clinical trials, and it's important to know which phase the treatment that you're being considered for might be at, because that gives you information about what studies have been done so far, sort of what kind of patients they're looking for, and different stages or phases will be recruiting different kinds of patients. As Dr. Mark was saying, initially phase 1 has people with more advanced disease, and then later phases, when we're looking for evidence of efficacy or effect, improvement in whatever we think the drug ought to do, tends to enroll people who have still better retained vision. So another thing that we'd like to discuss today is whether you want to decide first, if you find out that you're the kind of person that they're looking for a particular trial. First, you find out whether the trial is studying the disease you have. So that starts with going to a retinal specialist who can give you a careful and correct diagnosis and give you an assessment of what kind of vision loss you have and what kind of vision you retain. And then find out whether the treatment is something that you might want to participate in. And so one component of that decision includes whether many times patients want to be in a clinical trial because they want to get a treatment more quickly because there's not maybe a treatment that's already been approved for the disease that they have. So they think if they participate in a trial, they might be able to get access to a treatment that might help them that they can't get any other way. And that is true. It's possible that you might be able to get a treatment that might help you as a participant in a clinical trial. But there's probably an equal chance that you'll get access to a treatment that might not help you, in fact. And if they knew the treatment was going to help you, they would have already been able to report that and get the drug approved. So we're at a stage in time where we don't have a lot of treatments that have already gone through this whole approval process. So it's important to understand when you're considering participating in a clinical trial that you might get a treatment that might help you, or you might get a treatment that might not help you. And so it's important to do it such that I think one of the best reasons to participate in a clinical trial is because you want to advance the research. You want to help move things forward and help us better understand what's going on such that in the future we'll be able to identify treatments that may be helpful. And so knowing that there's a chance you may not benefit, it's important to consider the potential risks involved. And that might include risk in terms of safety, might also include risks that include things like inconvenience, time commitment, travel to a distant place perhaps, boredom, sitting through hours and hours and hours of testing. You heard Kathy B. talking yesterday about her participation in the Argus 2 study, and it was a job for her. Several days a week initially, she came and went and participated, not necessarily knowing whether she was going to benefit or not, but because she was committed to helping advance the research and the science. And also, it's also important to go in understanding that there is a chance that even if you go through a whole lot of testing and you get your heart set on being a part of a study, sometimes you go through some of the screening tests and find out you're not exactly the person they were looking for. You might not have exactly the kind of disease severity or disease manifestations that are necessary to participate in that trial. Or you might have another health problem that makes it impossible for you to participate in that trial. So sometimes we go through a long process of screening to determine that actually the treatment trial isn't quite right for you. So you have to go in kind of with a really open mind and a positive attitude, and have sort of expectations that you may or may not end up participating in a trial. And if you do participate, you should understand the risks include a lot of work on your part, a lot of commitment, and potential for not really benefiting at all. Although we all will benefit from the knowledge that is gained from the trial.
Right. And I think I would echo what Jackie said, and I find it very fascinating that people have very different outlooks on being involved in clinical trials. And I have some patients who are eager and want to do any kind of possible clinical trial is out there. And then I have other patients who wouldn't touch it with a 10-foot pole. And so let the other people try that experimental stuff. I'll wait around until we have a determination. And so I think a lot of it comes down to a personal decision. And additionally, I think the time commitment is a very big thing to consider. And for example, with our gene therapy trials, there are multiple visits, starting prior to the surgery. Then there's a surgery which is involved. There are multiple visits right after the surgery, and they start spacing out. But when you count them all up over the course of a year or two, it may be on the order of 10 or 15 visits. And if you live close to the site, that's easy, but oftentimes we have patients who are coming from far away, and that represents a pretty big commitment in terms of travel. And oftentimes those costs may be covered by the study, but it's still, a commitment, and it's a commitment for people who are helping you, because many of our patients need assistance in getting to where they need to go. I thought also we could then talk a little bit about just some of the different kinds of studies. And when you think about clinical trials, there are different flavors. So we can talk about longitudinal studies where we're looking at the natural history of the disease, and Dr. Michaelides, I think, can talk a little bit about that. We can talk about drug studies, gene therapy, and then also surgical devices. So maybe, Mike, you could talk a little bit about longitudinal studies.
For sure. So one of the ones that's very dear to our heart at the moment at FFB is the PROG-STAR study. So that's the progression in Stargardt disease study, where we aim to, over a 2-year period, get a deeper understanding of the natural history of Stargardt disease. So to be able to detect in a more timely, accurate fashion the rate of change using various different parameters. And the aim being, one, it will provide us more accurate information that we can then give to our patients to give them more information on their prognosis and how they'll do over time, but two, also in preparation for anticipated pharmacological studies, so drug trials for Stargardt disease. And to design that trial, you really need to have a really good understanding of the rate at which disease changes, to design them to be accurate and for them not to run for several years, because clinical trials are incredibly expensive and no one will fund a trial that might detect an effect over a 5-year period. You're really looking at 1 year to 2 years maximum. So that will be an invaluable study to help us plan for these Stargardt drug trials, which we hope to start in 2 to 3 years.
Yeah, I think longitudinal studies are very important, and one of the difficulties is that they are difficult to get funded, and that's where FFB has been very good about providing resources for that. We're also looking at a natural history study for X-linked retinoschisis, and one of the difficulties of looking at these diseases is that they do tend to be very slow. And when it may take you 3 or 4 years to be able to get a good measurable change, and the cost of that then is quite large. And so it is very hard to study these diseases in a formalized way. The other type of study is small molecules or drug studies, and I think some of you may have heard about the valproic acid study for autosomal dominant retinitis pigmentosa. That's something that our site has been involved in, and this is very similar to probably what most clinical studies are like. And in this particular study, we have a drug, we have seen in some models that it may have a benefit, and we want to try it in patients to see if there is a outcome. And so in our trial, what we're looking at is if we give patients valproic acid versus a placebo, do we slow down the degeneration? And how do we measure that? Well, we use visual fields as one of the main outcomes, and visual fields overall have been shown to be one of the most sensitive ways of measuring the progression of retinal diseases. Now, many of you have probably had visual fields and spent hours doing them, and they're not easy tests to do. And it's important to realize that for all of these tests, we can sort of divide them into subjective and objective tests. And it's very important to have a little bit of both. One of the things with clinical trials is that we want to get away from just purely subjective finding. You know, oftentimes you'll read something on the internet where somebody went and got some experimental treatment in another country and said, I tried this extract and my RP, I can see much better than I did before. That's a subjective finding. And there are many reasons that you can have subjective improvement. To get an objective finding, we need to look at other things like structural changes that we can measure, things like the electroretinogram, and these things are independent of that kind of change. And so, for example, in a drug trial like valproic acid, we're doing a little bit of both. We do visual acuity, which is a subjective measurement. Even visual fields have a subjective component to them, but we also do things like electroretinogram and optical coherence tomography, or OCT, which is a structural measurement, and Jackie can talk about adaptive optics, which is another structural measurement, and these are objective measurements, and those are very important as well.
Right, so by subjective, a subjective measurement is one where the patient tells the doctor what they experience. So they say, they read letters on a chart, or they push buttons when they see lights, and they report to us what they're experiencing. So that's very important information that no one else can provide. Objective measures are ones where we can make a measurement without, a doctor can make a measurement without any response at all from the patient, if the patient's cooperating with the test. So examples that Mark was giving were like an electroretinogram, where we put, it's a little bit, most of you may have had this test before, it's analogous to an electrocardiogram or an EKG test, where you put a sticky electrode on the chest and measure electrical activity that the heart makes whenever it beats. We put a little contact lens with an electrode on the cornea on inside the eyelids, and we measure the electricity that the eye makes whenever it is exposed to light. And you don't have to say, yeah, I see that, or no, I don't see that, or what it feels like to you, or what it looks like. We are able to make those measurements without any response, any voluntary response from the patient. So those are that's how you define sort of the difference between subjective and objective. In addition to electroretinograms, which measure the way the retina functions in response to light, we can use a picture of the retina called OCT that uses sort of a low energy kind of light to make a picture of the retinal layers in cross section, sort of a slice of the retina. And that is a nice test because it doesn't touch the eye at all and gives us information about the different retinal layers and how they're doing in response to disease as it gets worse or in response to a treatment. That test can also measure some signs of side effects like swelling in the retina that can sometimes limit vision in response to treatments. And then we do some additional imaging techniques that are special pictures that enable us to actually see the individual vision cells themselves using special techniques called adaptive optics. And all three of us up here have had experience using those novel sort of high-resolution imaging technologies to get really good high-resolution pictures of the retina. These are the kind of pictures of the individual retinal cells that are analogous to the kinds of studies that are done on animals that are treated with different treatments. But in the animals, we can only get these kinds of pieces of information when we take the eyes out and cut them up and look at them under a microscope. So these additional non-invasive high-resolution pictures that we can take in living eyes are really helpful in helping us better understand what's going on with the vision cells as the disease gets worse and in response to therapies. So maybe we should talk about some different kinds of therapies in addition to drug studies. Drug studies are helpful because you can sometimes take a pill or use an eye drop, and it doesn't necessarily require any direct intervention in the eye. Whereas some of these other additional kinds of treatments we're going to talk about are a little bit more directed right at the eye.
So gene therapy, I think, has been one of the most exciting things that has happened in the past decade for retinal degenerations. And this, of course, started first with Leber's congenital amaurosis, which is a rare form of retinitis pigmentosa that's very severe, affects children. But now we're moving on to other diseases as well. And currently we are involved in a gene therapy trial for Stargardt's. And Stargardt's is caused by a mutation in the ABCA4 gene. And what we're looking at is a virus that is carrying a normal copy of this gene. And the concept is essentially to deliver that virus into the eye, where it will go into the retina and actually bind to the photoreceptor cells that are missing this protein. It will deliver that normal copy of the gene and replace it. Now, one thing I think that's important to realize about gene therapy, at least at this point in time, the hope would be to really slow down the disease, stop progression, to sort of buy time for another type of therapy that could then replace vision. Gene therapy potentially could help vision a little bit. If you had sick cells that are not functioning well, it might improve their function, but it's not going to bring back cells that have already died. And so really, ultimately, we're probably going to need more than one type of therapy for these diseases. But gene therapy is something that can fix the defect, buy some time, maybe until some other modality such as stem cells comes along. You know, these studies are very intensive, and patients need to do a lot of testing for it. It does involve a surgery, which always brings risk. And so you can have an infection, after surgery. So there's always potential of losing vision. But once again, we start with people who have very severe vision loss. We're very careful. And one thing that's important about clinical trials is that we do it in stages. And so we'll treat a few patients, and then there's usually something called a data safety monitoring board that is an independent group of experts, both doctors and scientists, who will look at all of the data from the first couple of patients tested and then give the approval to move forward. And so at every stage, outside sources are checking to make sure that the treatment is safe and that it can progress along. And this becomes even more important when you're dealing with even more invasive things, such as a surgical implant, and Jackie has a lot of experience with that. So.
It's interesting he should say that, because I don't actually do surgeries, but I do have experience with everything but the surgery for a couple of trials that are treatments that are administered using surgery. So one of them is for a device that releases a protein into the eye that is supposed to act a little bit the way gene therapy does, that slows down the progression of the vision loss through keeping the cells alive longer. And that is through a protein known as ciliary neurotrophic factor, or CNTF. Because it's a protein, if you put it in the eye, it doesn't last very long. If you just put it in there, it'll be gone in a few days. However, this company called Neurotech designed a little device, which is about as long as a piece of rice and about a quarter as thick, that gets put into the eye with one surgery. And the surgery itself takes about 15 minutes. It's not a very hard or complicated surgery. Once the device is positioned in the eye with the surgery, it releases the protein, CNTF, into the eye over many, many months to years, and continues to produce this protein that is believed to, and we hope will, keep the cells alive longer, such that the disease progress is slowed. In that particular trial, we do a sham surgery on the other eye, where we don't put anything into the eye, but we do a surgery on the surface, so patients are masked to which eye got the treatment, hoping to make them less biased toward the results of the trial. Another surgery was one that you heard about yesterday, where unlike the treatments like gene therapy and CNTF, which are designed to slow vision loss, the Argus 2 trial was of a device that is the first that's been approved by the FDA for restoration of vision, restoration of sight for people with advanced vision loss. And I think you heard more eloquently about that trial than I can ever convey to you from the participant, Kathy B. She's at the Argus 2 booth, or the second site booth, if you wish to speak with her later this meeting. But in that trial, much more complicated surgery is performed, where the device itself, which is about half as big as a pinky fingernail, is placed onto the surface of the retina, in the area right in the center, which we all use to see fine details, which we use, it's called the macula, which we use to see the very best vision that we have. So that device is placed right in the center of the retina. It is connected through a cable to a device that sits outside the eye but under the skin of the eye, called the conjunctiva. And that surgery takes about between 2 and 4 hours to complete. But as you heard yesterday, the healing time for that is a few weeks. And after a few weeks, people who have this device in their eye really don't have any discomfort and are not really aware that they've even had really surgery on their eye. After several months of fitting and rehabilitation, I kind of liken it to when a person gets an artificial leg or arm, they need to go through rehabilitation to understand how to make use of that new limb. This is a prosthetic retina, and so initially when you get that device, you don't really understand how to interpret or use that information. But through sort of a lot of work with bioengineers and low vision specialists and rehabilitation counselors, people are able to really understand what the information that the device delivers to the eye means, and they're able to interpret the information in ways that can really help them see a lot better, as you heard yesterday.
All right, well, I think we've talked enough, so I think we should go ahead and just open it up to audience questions.
So, because we're recording this session, can you wait until a microphone?
Sorry, Jackie.
I was just going to say
Okay. Thank you. The implant done by Neurotech that was just discussed, is that still in the clinical trial stage? And if so, at what stage?
Great questions. So that is in a clinical trial. It's in a phase 2 trial. So the story of neurotech is a long one. They started with a phase 1 study back in the early 2000s where they implanted 6 patients who with very advanced vision loss for a period of exactly 6 months, at which point the device was removed. During that 6-month period, they observed some vision improvement in people who could not even see the big E on the chart after the device was placed in their eye, which was a really promising result. So those devices were removed because that was the plan of the study. And the promising results from that phase 1 study led to the development of a larger multi-center, which means many different clinics across the country participated, phase 2 clinical trial. It was actually granted phase 2/3 status because it's for a rare disease, suggesting if the results were really promising, they could perhaps use that as an early phase 3 study. The other thing about phase 2 is it's a dose escalation study too, so you kind of have an opportunity in the phase 2 studies to study 2 different doses. So in that larger trial of many that was done in many centers between 2007 and 2009, actually had 2 trials, one for people with more advanced RP, where they were looking at visual acuity over 1 year as an outcome. And then they had another trial for people who had really early retinitis pigmentosa and really good visual acuity, where they measured side vision, visual field, over 2 years. And they observed those patients from 2007 to 2009. So the results of that trial suggested that, and these results just came out last month, suggested that the eyes that were assigned to receive the high dose of the CNTF over time, and this is perhaps the way the treatment works, had actually not as good side vision as the eyes that got the sham treatment. And the thought there is, was observed in animal models too, so it was not unanticipated. Thought there is that it slows down the death of the vision cells and keeps them alive longer by making them a little less active, a little less sensitive to light. There was a low dose in that group where that effect was not appreciated. As a part of this trial, studied about 120 people, and there were no significant safety concerns, anything like that. We had an opportunity to study 3 of the people in the early stage disease trial with people with very good vision and just measuring side vision loss using this new technique we were talking about where you take pictures of the vision cells in the eyes. And we saw that the people who got either the high dose or the low dose had more vision cells surviving at the end of 2 to 3 years than their opposite eyes, which got the sham treatment, which makes us think that the treatment did keep the cells alive longer. However, nobody wants more cells alive that can't see very well. So we're currently doing a study, which is phase 2, at UCSF, where we're looking at the low dose implant to see, using the special pictures of the vision cells and following those patients over time to see if we can keep the cells alive longer without having a reduced visual field effect.
Thank you. You answered my second question, which was there objective data or only subjective data? And I understand now that there was objective data. My final question is, can a patient enter a clinical trial at different phases, or do they have to begin at the beginning?
That's another great question. So most of the time, clinical trials are listed, actually we should mention this right now, there's a website called www.clinicaltrials.gov. It's all one word, clinicaltrials.gov for the translators. That will list all the clinical trials that are currently happening for every disease. So if you type in your disease and where you live, or sometimes just your disease, you will see a list of all the clinical trials. Some of them have been completed and some of them are still recruiting. Most of the time, a clinical trial will recruit patients until they meet their enrollment quota. So most of the time people have a protocol and they say, we need to enroll this many patients to identify the effect we want to see. That's going to give us confidence that it's a true effect. So after that quota has been met, then they continue the trial, but it's close to further enrollment. So you want to look on that website for trials that say recruiting. And oftentimes they think they have a little difference in font color, where the recruiting ones are in green and the ones that are close to further enrollment will be in red.
And the other thing would be that for the different phases, oftentimes they're looking for different levels of severity. So if you wanted to be in a phase 1 trial, you may not necessarily qualify for some of the other phases. So they're going to have specifications. And additionally, oftentimes when you decide that you want to go and be in one of these trials, you'll often have what's called a screening visit, where they will do some preliminary testing and see if you actually qualify, because sometimes we'll have criteria, saying that your visual fields must be at least 10 degrees to qualify for this particular trial. Or in other cases, we might say that, well, your visual acuity has to be 2200 or worse. And if you don't pass those, even though you may fit the criteria, you would not be enrolled in the trial. And oftentimes we have to test many more patients than would actually be enrolled in the trial to get people who fit all of the criteria. And when you look at the criteria, sometimes it's 18, 19, 20 different exclusion criteria.
Wait, let's get you a mic. Where's the mic runner? Oh, there you are. Okay.
Yeah,
So,
she has-
if you had a question, she can go. That's fine. Whoever was next, you could go.
Do you anticipate the new registry system changing the enrollment process?
That is a really great question. So the registry that is sponsored by the Foundation Fighting Blindness is just rolling out, and I think so it hasn't really been fully implemented yet, but my understanding is the intention is to serve as a resource for just exactly what you're talking about, so that investigators who have a potential treatment that they want to study in a particular subset of patients can work with the foundation to identify people in the registry who have identified themselves as people that want to participate in trials, have given their permission to the foundation to be contacted when opportunities arise, and it should really accelerate the ability for clinical trials to enroll patients that have some of the right inclusion criteria. So we're hopeful that's going to be a really valuable resource for the vision community.
Yeah, absolutely. And I think something we kind of hammered at last year at Visions was the value of genetic testing and actually knowing what your mutation is. And I think I would sort of reiterate that the more we know about you, the better we can match you to a clinical trial. And the cost of genetic testing really just keeps coming down every year. It's like computers. Every year you get more. And so it's actually really affordable for many people to go ahead and do that. And even if it's not, there are other ways to get genetic testing done. So I would encourage everyone to really look into that.
Hi. So my 12-year-old son has Stargardt's, and he's being treated by Stephen Sang in Colombia. So he did mention in his last visit that he could come back in 6 months and maybe discuss drug therapy. So number one, could you talk a little bit about that? And number two, when they look for safety in the drug therapy, I know that you said they look for things in the eye, but do they also look at other parts of the body
Absolutely.
to make sure they're not being affected?
Yeah. So first off, in general, when we do clinical trials, we're not just looking at the eye. We're drawing blood. We're checking liver function tests. We get urine. We look for antibodies. For example, when we do gene therapy, we test the blood for antibodies to the virus to see if the immune system is mounting some kind of response to it. And then just depending on what the treatment is, we'll tailor it to that. For the valproic acid study, one of the known side effects of valproic acid is that it can raise your liver enzymes. So that's something we check routinely in patients. So for every type of study, there is going to be a whole host of safety tests that are monitored. And the frequency of those are going to depend on the actual therapy itself. In terms of Stargardt's, I'm thinking that in terms of the drug, I'm thinking they're talking about a visual cycle inhibitor. Yeah, fenretinide. Mm-hmm. Mm-hmm.
Oh, sorry. No, he just mentioned something about a vitamin A substitute, I guess, and it was just very quick at the end of a visit, and so he didn't even tell me the name of it. He just said, come back in 6 months to discuss it. And was worried about doing that since he still has some pretty good vision left.
Yeah, I mean, there are trade-offs, and I think these visual cycle modulators have been around for a while, and it sort of gets back to what Jackie was talking about with CNTF, is that there is some evidence they may reduce your dark adaptation ability at the trade-off of preserving your cone cells. And, you know, for some people, that trade-off is worth it. For other people, I have some patients who say, you know, I already have a hard time getting around in the dark. I don't want to take anything that's going to make my night vision worse, even if it's going to preserve my central vision. And I have other patients who say, yeah, I'll try it. I think a lot of that sort of comes down to a personal decision.
Other questions?
Hello. I read earlier in the In Focus magazine put out by the foundation, about a possible clinical trial in 2015 for RP, where since they talked about the cells before they die, they go dormant. And do you all know anything or involved in any of that? Trying to reactivate the dormant cells.
What you might be thinking of, if it's not something that's already underway, so it's thought that maybe CNTF can actually keep alive cells that are not quite gone but are sick and on their way out, but that wouldn't be something starting in 2015. What you might be thinking of is there is RDCDF. There's a trial of a compound, a protein that was discovered by Dr. Jose Sahel, who was here at this meeting but had to leave. Isabel Odo, his collaborator, is here still, Dr. Odo, AUDO. They have identified a protein that is produced by rods and keeps cones alive longer. And so it may be that kind of protein can activate cells that are not well, that are sick and on the way to dying, and better enable them to see. The other thought would be there are these optogenetics treatments where they're trying to get cells that haven't died yet to, or may have lost some part of their ability to see. So for example, a dormant cell might be one that's lost its outer segments, the part that turns light into a message your brain can understand. Turn them into, restore the ability to perceive light to those cells. And that's through a field called optogenetics. And those trials are sort of in the very sort of earlier preclinical stages. They might be ready for a clinical trial by 2015. So that might be what you might have heard about, but I'm guessing. Yeah. One take-home message while we're getting the microphone to the next person is there's so much exciting work going on. I mean, I've been coming to Vision since I think maybe 2007 or maybe 2006. And in the beginning, it was really all about mouse models and designing mouse models and treating mouse models. And patients were pretty sick of hearing how good we were at treating mice. I mean, they were just ready to have people be participants in clinical trials. So I think the fact that we are sitting here with you today discussing all these clinical trials that are underway or about to become underway is just an amazing advance over the last 5 years. It really is. Yeah. Thanks to the
Yeah, I have kind of a practical question. I think Mark alluded in his remarks to the fact that there are times where there's funding for travel, and I'd like to explore that a little bit because that can be a very important issue to people, particularly if there are repeated visits and they have to travel a distance where there's a hotel and an overnight stay because of the distance they travel, airplane fare perhaps, if you don't have a multi-center study. My question is, I mean, in general, I want to get a sense of how often the study provides for funding. And sometimes it's not only the individual, but it could be a family member that would have to come along with them because of the nature of their condition. They can't really travel comfortably by themselves. So I guess practically speaking, what would be the expectation or the range of expectations for an individual that might be considering participating in a study where they have to travel an extensive distance and have frequent travel?
So I can tell you, we always ask for travel funds, but when you have a clinical trial, I guess there's another thing we haven't really emphasized, that there's generally a sponsor of that trial. and they are the ones who are paying for the clinical trial. So in some cases that may be a company. For example, with some of our gene therapy studies, there's a company called Oxford Biomedica. In other cases, it may be FFB that's sponsoring the trial. And in some cases, it may be an NIH funded trial. And so when you start these trials, those are things that are usually negotiated for at the beginning. And of course, as a site, we always want them to give us travel money. But what will happen is usually they'll come back and say, okay, we'll give you X amount for travel. And in some cases, it turns out that that's not enough. And I can think of one trial that we have where we had only recruited 2 patients, and the sponsor came to us and said, you guys have been doing this for a long time. Why do you only have 2 patients? And we said, well, because you didn't give us any travel money. And they said, all right, well, we'll give you travel money. And within a month, we had 10 patients. And so I think they, somewhat it's education as well, telling the sponsors how important that is. And it was sort of a similar thing with the valproic acid study. They had a limited amount of money for travel, and recently they increased that enough to actually have someone come along with the person, and that actually has boosted enrollment. And I think that the organizations and FFB especially are realizing that that is a very important component, because there aren't enough centers out there to have one in every geographical region for people. And if you have a rare disease, chances are you are going to have to travel somewhere, unless you're lucky to be located in that city. But certainly we always love it when they have travel money. I wish that would always be the case.
Another motivator to get us all excited about raising funds for foundation funding plans.
I actually had sort of a twofold question. Have you heard anything about saffron supplements in clinical trial? And also, regardless of what drug therapy you do, if liver enzymes, for example, were not good, you withdraw the drug and the liver enzymes go back to normal, or do you not know that?
So I think the first question about saffron, my good friend Benedetto Falsini in Italy is doing a trial with saffron for Stargardt's, and I think that's underway. Haven't heard any results of that, but that is something that is being studied. Benedetto Falsini, he's at the Catholic University, Falsini, Falsini. In terms of the liver enzymes, a lot of it's trial dependent, and I can just talk about the one trial we have right now with valproic acid. Oftentimes, you can get elevated liver enzymes for a variety of reasons. If you just have a cold, a virus, your liver enzymes will bounce up, and so usually those will resolve. And so when we have someone in a trial and we see an elevation of the liver enzymes, the question that comes is, well, is it really due to the drug, or is it just because they had a cold? And so oftentimes what we'll do is we'll stop the drug, wait until the enzymes go back to normal, and then you can sometimes re-challenge with the drug. And if the enzymes stay normal, then you know it was not the drug. If they go back up again, chances are it was the drug, and you need to stop it.
Okay. You had mentioned the website www.clinicaltrials.gov. Would that website include trials that might be done overseas, out of the country as well?
So I think if any clinical trial that gets NIH funding is required to register on clinicaltrials.gov. And I think most journals these days, if you want to publish your results in a journal, it needs to have been registered. So it's going to include the majority of trials. I mean, there may be some that are not included, but if they're not on clinicaltrials.gov, I would be a little,
There are some other registries, so and
Yeah.
Mike may know more about them. There's an Australian one. There are a few other ones. So one way to access ones besides clinicaltrials.gov, which, as Mark correctly stated, almost every trial in the United States would certainly be listed there. If you go to genetests.org, genetests.org, and type in your gene, sometimes that will have information about trials that are ongoing, and that will list anywhere in the whole world that the trial might be ongoing. So there's a lot of resources, and Foundation Fighting Blindness might be a way to find out if there's more than just what's listed on clinicaltrials.gov, but that's certainly a good starting point, unless you have your own Learjet and you want to fly somewhere else in the world to participate in a clinical trial, in which case you might want to be more comprehensive and look at all sorts of, are there some other registries?
There's a European one, so
Yeah.
the European equivalent to clinicaltrial.gov.
And there's an Australian one, I believe,
Yeah.
and
Yeah,
so there are a
there
number
is.
of other ones similar. But clinicaltrials.gov will also, so oftentimes there will be, for example, the Argus 2 study had centers in the United States and in Europe, and all of those centers would be listed on clinicaltrials.gov, even though it's primarily an American trial, right? So the French and the German, different sites would also be listed there.
I think we've got time for one or two more questions.
That would be me, I think.
It would indeed.
Me.
One more.
One in the back, yeah.
Oh,
After you.
Oh, okay.
after me, okay. Since you brought up the neurotech CNTF trial, actually there's two things that I wanted to ask about. I tried out for that trial, and this is like the story of my life. I had a little too much vision for the end stage, and I had not enough for, I just fit in between. And so that's been one of my frustrations, is that I just feel like I'm in between and I don't qualify for anything. So I'm hoping that at some point, that there's going to be some trials that, that many of us that didn't qualify for, will. I have another one regarding the neurotech as well. When that neurotech trial first, we were all excited about it, and I know that they started off with the CNTF as the first neurotropic factor to be looked at. But there were a whole bunch of other neurotropic factors that they promised us were coming down the line. Are we anywhere with those other factors? Because I've heard nothing.
Right, that's a great question. So I worked during my Career Development Award years with Matt LaVale, who a Foundation Fighting Blindness, trustee in the past, scientific advisory board member, had discovered over many, many years that there were a lot of different growth factors that helped keep cells alive in a number of different animal species. Well, the problem is that CNTF, neurotech had to sort of pick one to go with. They chose CNTF because they were able to get rights to that protein, and it had the advantage of having been demonstrated to save photoreceptors in 14 different animal models spanning 4 different species. So it looked to be the one that was going to be most broadly applicable to people, regardless of what their mutation was. That was the reason that they chose that particular protein and went forward with it. And so, yeah, there's certainly a lot of literature suggesting there are other growth factors that might be helpful in keeping photoreceptors alive. Neurotech did not choose to pursue anything besides CNTF, to my knowledge. I don't work for them, and I'm not a consultant for them. I don't get any money from them at all. They do provide us devices. But my understanding is that is the only photoreceptor protective growth factor they have chosen to study. And so if that answers your second question, to get back to your first question, yeah, I have had a number of patients who also kind of fell within the gaps. And I think the thing we learned from those earlier trials is that if CNTF works, it works in early disease. It works in people who have a lot of cones left to save. There is probably a window of opportunity beyond which it is not very effective. So for the people with the more advanced stages, it was not very effective. So you can consider that in some ways a blessing in disguise that you didn't participate in a trial that might not have benefited you. However, it's frustrating to not be able to be a part of something you want to participate in and contribute to, and we commend you for wanting to be a part of science. And I'll tell you that as time goes on, I promise you, there will be something that will be just right for you. The beautiful thing about neurotech is that it was sort of designed to be non-specific, and it didn't matter what your genetic mutation was. And so it appealed to and may be relevant for a number of different types of mutations. until the next thing comes along that is like that, right? So valproic acid is just for autosomal dominant RP, and gene therapy is just for the people who have mutations in the gene that they're studying. There's a lot of research going on towards development of things that will be more broadly applicable, and that includes drugs like small molecule therapies that might improve folding, might reduce oxidative stress. There are things coming along, and as your disease progresses, you may be eligible to participate in an earlier phase than you were before, but you still will be likely to participate in something. I really believe that you should hold out hope and keep looking at that clinicaltrials.gov website, because I think if you hang on, I promise you someday something will come along. Okay, I think
yeah, it's recorded.
So actually, we're looking, in my lab, we're looking at another growth factor called BDNF, and we're actually looking at ways to upregulate its expression via serotonin receptors. And the really interesting thing about this is that we already have a whole host of drugs that modulate the serotonin receptors, like the antidepressants. And so if you look at antidepressants, it actually increases BDNF in the brain. And what we're trying to do is elicit that effect in the retina to try to get neuroprotection of retinal cells. So I think that's another, we're not just looking at CNTF, there are lots of other things that are happening as well.
Super. Should we take the last question at the back?
One quick question.
I think there was a gentleman.
This guy has a mic.
Okay.
Quick question on the RP or the protein delivery clinical trials you're talking about. Is that just for RP, or is that going to be including other genetic diseases like cone rod?
Are you talking about CNTF? Yep. CNTF. So the CNTF studies that we're doing right this minute are just for RP because I'm trying to make it a sort of homogeneous patient population. However, CNTF was recently, in the last year, also studied in patients with achromatopsia because of animal model data in a dog that suggested it improved function of the photoreceptors. So it is not inconceivable that someday it might not also be used for cone rod. Right this minute, it's just being restricted to RP.
Weren't there some choroideremia
And there were a few choroideremia patients in the original study who didn't appear to fare any differently than the RP population. So again, it might be relevant for people with lots of different diseases. Let's hope it is.
Okay, last question.
Okay. Just very briefly, in terms of trials that are going on in the United States, I'm from Canada. Can I qualify to be, provided everything else is correct?
Yes.
Do I qualify?
Yes.
If you're willing to come to the trial site, yes.
Yes.
I mean, right now I have a patient from Brazil who wants to come and do one of our trials, and that's a much longer trip. So.
Well, now, which beach are you near when you say, am I willing to qualify?
Ocean Beach, San Francisco.
I'll be there.
Come on
So.
down.
Thank you very much.
Okay, that's super. Thank you for attending this session. There's a grab-and-go beverage station set up in the Grand Ballroom foyer until 3:45. So if you'd like a drink on your way to the next session block, which is the popular Doctor is in Q&A beginning at 2:15, please check your program for disease groups and locations. Thank you very much.